Hi everyone, this is topic 8 of the human anatomy class and this is the third lecture on nervous system. In this lecture we are going to learn about the transmission of nerve impulses. There are 4 stages in this transmission. The first stage is called resting neuron, and from the name itself you know that the neuron is resting. So the plasma membrane of the neuron at rest, it is said to be in a polarized state, and the voltage of the plasma membrane is called resting membrane potential. Broadly, at this stage, the plasma membrane has more positive ions outside than inside. That means the inside is less positive. What kind of positive ions are there? Sodium ions, which is denoted by Na, and potassium ions, which is denoted by K. So both of these are positively charged ions. Concentration of sodium ions is higher outside the cell and the potassium ions are more inside the cell. The second stage of nerve impulse transmission is called depolarization. In this stage, a stimulus comes and is said to cause depolarization of the neuron's membrane. In this stage, the membrane of the neuron becomes permeable to sodium or Na positive ions because the sodium channels open. This is passive transport, and the depolarized membrane allows the sodium ions to start flowing in passively. The movement of sodium ions starts an action potential in the neuron, and in the place where the inside of the membrane is more positive, it produces a graded potential or a localized depolarization. If the number of sodium ions entering the cell is above a certain threshold, then the action potential starts traveling, or it is propagated over the entire length of the neuronal axon. And these impulses or action potentials, they travel faster if the axon is covered by a myelin sheath. The third stage is called repolarization. In this stage, potassium ions, which are also positively charged and denoted by K positive, they start rushing out to restore the inside of the membrane to a negative charge and the outside to a positive charge. This is because the entry of the sodium ions had made the inside more positive. So this is known as repolarization. Sometimes hyperpolarization occurs. That means more potassium ions go outside than the amount of sodium ions inside. The fourth and the last stage of the transmission of nerve impulse involves the refractory period. In this phase, the initial ionic conditions, that means what was present during the resting membrane potential, is restored, and active transport is used for this purpose. Sodium potassium pump is used. This pump uses ATP. So ATP is broken down. For every one molecule of ATP that is broken down to release energy, 3 sodium ions are kicked out from the cell and 2 potassium ions are brought inside. This graph shows the action potential in a neuron. If you look at the y-axis, it denotes the membrane potential in millivolts. Anything above 0 is positive, below 0 is negative. The x-axis shows the time in milliseconds. At the beginning of the graph, it is the resting membrane potential. You see the membrane potential is at baseline. Then at around 0 time, the membrane potential starts increasing. It reaches the threshold potential and then it gets more and more positive. As the graph is rising up, it is depolarization. That means sodium ions are moving in. After a maximum value, the graph starts going down. This is repolarization. This is where potassium ions are moving out to make the outside more positive again. Sometimes hyperpolarization may occur. You see the graph dipping even below the baseline, below the resting membrane potential. And finally, the refractory period, the resting membrane potential is restored. This graph shows how the different channel gates help in the 4 stages of the action potential. This figure shows the propagation or movement of the action potential. If you look at the figure on the left side, a tiny portion of the axon, the neuron has been shown in yellow color. So you see the tiny portion of the axon is enlarged here. So below that axon, action potential, that means localized depolarization, is shown with purple color. You see that the ionic distribution, it has become more positive inside the membrane than outside. This is because sodium ions have entered, and this is only localized, okay, at a certain location. Then gradually the action potential is moving in one direction. It is moving to the right side. So the purple portion has moved, and the portion which was purple before is now in repolarization. That's because potassium ions are moving out of the cell to the outside. Then in the third phase, in the third part of this figure, the action potential has moved further, and the repolarization has also moved right behind the action potential, while the first part, which was purple in the beginning, is now back to the resting potential. The figure on the right side is also explaining similar things. When the action potential reaches the axon terminal, either vesicular or non-vesicular synaptic action occurs. This figure shows a vesicular synapse. This figure shows neurotransmitter molecules diffusing across the synaptic cleft containing the synaptic fluid. Neurotransmitters will eventually generate an action potential in the postsynaptic neuron. Another image of a chemical or vesicular synapse. Vesicular synapse again. Reflex arc. Reflex is a rapid, predictable but involuntary response to a stimulus or trigger. And this occurs over specific pathways called reflex arcs. A reflex arc consists of a direct route from a sensory neuron to ultimately an effector neuron via an interneuron. There are 5 basic elements or parts of a reflex arc. The first one is a receptor, which is a sensor. For example, a sensor on the skin. If you touch something hot, it's the sensor that detects it. Then the signal from the sensor is taken by the sensory neuron. Here shown in turquoise blue, the sensory neuron takes the message from the sensor to the CNS. The third element is an interneuron or an association neuron. This is present in the CNS. So here it is shown in dark blue. So the interneuron is receiving the message from the sensory neuron, and it will relay the message to the motor neuron after the message has been processed. So the motor neuron is the fourth element. It is shown in red here. The motor neuron takes the message from the interneuron back to the PNS, to one of the effectors. And finally, the fifth component is the effector organ, which is usually a muscle or a gland. There are 2 types of reflexes, somatic reflex and autonomic reflex. Somatic reflexes stimulate voluntary skeletal muscles. For example, when you touch something hot, you pull your hand away. Autonomic reflexes control the activity of involuntary organs such as smooth muscles, heart and glands. This could be, for example, you see something scary and your heart starts beating faster. Your smooth muscles, after digestion, they are doing peristalsis. All of these are autonomic reflexes. This is a list of important definitions that I had asked all of you to memorize in the previous lecture. These are nuclei, ganglia, tracts versus nerves, what is gray matter, what is white matter. Now we are going to learn about the human brain. There are 4 regions in the human brain. These are the cerebral hemispheres, also known as the cerebrum, the diencephalon, the brain stem, and the cerebellum. Inside the brain, there are also some hollow spaces, empty spaces, which are called ventricles, and these are filled with a fluid called the cerebrospinal fluid, or CSF, whose composition is very similar to blood plasma. This is the adult human brain with all the 4 regions. The most superior portion of the brain consists of 2 cerebral hemispheres, which are collectively called the cerebrum. The cerebrum is responsible for the brain's cognitive functions, including learning and language, conscious interpretation of sensory information, conscious planning of movement, and personality. Its surface consists of elevated ridges called gyri and shallow grooves called sulci. Deep grooves called fissures separate major regions of the cerebral hemispheres. For example, the longitudinal fissure separates the right and the left hemispheres. The central sulcus separates the frontal and the parietal lobes of the cerebrum. This figure shows the cerebrum. The cerebrum is divided into 5 lobes by fissures. These lobes are the frontal lobe, parietal, occipital, temporal, and deep insular lobes. This figure shows the lobes of the cerebrum. This is the photograph of a real human brain, and look at the different lobes. Cerebrum contains several specialized areas. For example, the primary somatic area located in the parietal lobe receives information from the body's sensory receptors. The primary motor area is located in the frontal lobe. This sends impulses to skeletal muscles. Broca's area is involved in the ability to speak. This is located in the frontal lobe. This figure shows the different specialized areas. This slide lists some other cerebral areas which are involved in special senses or interpretation. For example, the gustatory area, which is for taste, visual, auditory, olfactory. Olfactory means the sense of smell. The interpretation areas include language comprehension or general interpretation. This figure is pointing out again the olfactory area, language comprehension area, and some other areas. The cell bodies and unmyelinated axons and dendrites of the cerebral neurons lie in the cerebrum's outer 2 millimeters in a region called the cerebral cortex. These portions of the neurons are unmyelinated, which gives the cerebral cortex a gray color, and for this reason it's called a gray matter. The cell bodies and processes of the cerebral cortex communicate with other parts of the nervous system by bundles of myelinated axons, which is called the white matter. It's because the myelin sheath is white in color. The largest tract of cerebral white matter is called the corpus callosum, which connects the right and the left cerebral hemispheres, and it allows interhemispheric communication. Another prominent tract of white matter is inferior to the corpus callosum. It is known as the fornix. However, the gray matter isn't confined to the cerebral cortex only. There are clusters of cell bodies called nuclei which are found throughout the white matter of the cerebrum. An important group of nuclei, the basal nuclei, monitors voluntary motor functions. The neurons of these nuclei are connected to other parts of the nervous system by various tracts of cerebral white matter. This figure points out the cerebral cortex containing the gray matter and then the white matter deep inside.